In this video lecture, we will talk about how enzymes speed up metabolic reaction like this one. So an enzyme is going to interact with the substrate, they're going to form a complex, and as a result, the chemical bonds between the substrate is going to break and you have to have two products. So let's start. I'm going to first start talking about the characteristics of enzymes. Then I'm going to talk about the factors affecting enzymes, and then I'm going to talk about the activation energy. Later on, in the other part of the class, I will talk about cofactors and about enzyme inhibitors. So now let's start talking about these three main things.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
Okay, let's start. And here what you can see is a disaccharide that is called sucrose. So you have a glucose joined by a covalent bond with a fructose. And when you add water and the enzyme sucrase, what is happening is that the glucose take the OH and the fructose will take the hydrogen, and now you have two products. In this case, this chemical reaction happened very fast, and the sucrase was able to break this covalent bond here. And this chemical reaction can happen naturally, but it will take a long time. When you have an enzyme, you are able to catalyze this reaction very fast in terms of seconds, and the enzyme, the sucrase, is not consumed as a result of the chemical reaction, but it will be regenerated and it can be used again and again. So there are a few things that I want that you remember: that a catalyst is a chemical agent that speeds up reactions without being consumed by the reaction. So as I told you before, this kind of reaction can happen randomly, but it will take a long time. However, when you have a chemical agent like an enzyme, like the sucrase, this chemical reaction can happen very fast and it can happen without being consumed. An enzyme is a catalytic protein, and in the case of the hydrolysis of the sucrose by the enzyme sucrase, this is an example of an enzyme catalyzed reaction. So remember 3 points: catalysis, that an enzyme is a catalytic protein, and third, I show you the hydrolysis of sucrose, and this is an example of an enzyme catalyzed reaction. I'll show you this figure to explain what is an activation energy that we have discussed before, but remember that most of reactions don't start spontaneously. They require energy such as spark to get started, and this is what we call an activation energy. So far, I told you that enzymes are biological catalysts that increase the reaction rate of biochemical reactions. Now I would like to show you some of the characteristics of enzymes. And to do that, I wanted to show you this model that you see here. And this model is a model of a protein, and this is an important trait of the old enzymes. Enzymes are mainly proteins, Although recently it has been discovered that certain molecules of RNA can also have enzymatic properties. So this is something that is important. Another property, another characteristic of an enzyme is that they are very specific. Yeah, so they have only certain set of substrates that they can fit in their active site. So imagine this is the active site and they will have a specific shape. Are only molecules that have this specific shape. So if you have something that looks like a circle, something that looks like a triangle, something that it looks like a square, it's not going to fit here. Yeah, those one is not going to fit. The only one that is going to fit is one that have this very weird shape that I just made. So enzymes are very specific. And finally, another trait that is very important of the enzymes is that they can be used over and over. For example, this substrate that I show here goes here and attach into the active site. Later, when it produces maybe the two products, later the enzyme is going to can be used again and again. Okay, so let's recap all the things that I just told you. First, I told you that the enzymes are made of proteins. Yeah, although recently they have been found that RNA molecules they can also be involved in enzymatic properties, and these studies are very interesting because they talk about the origin of life. So possibly RNA in the beginning has also enzymatic properties, and it's something that is still under study, but is fascinating. Another important characteristic of the enzymes is that they are very specific and they only work with certain reactants or substrate. And I explain you, and later we will explain more about what is an active site and how they fit. The substrate they can fit in this active site. And finally, I talk about that enzymes can be used and you use over and over again. So now I'm going to explain maybe in a more detail what happened when the substrate interact with the active site. So here you have in this figure you see an enzyme, and when the substrate goes inside of the active site you have a enzyme substrate complex, and this will produce a conformational change in the protein that will allow the enzyme to interact better with the substrate and allow the reaction to occur in an easier way. And this is called an induced fit. So remember that the enzymes bind with a substrate and that this causes a change in shape of the enzyme. And also remember that this change in shape allows the reaction to occur. And finally, that this is called induced fit. This is a diagram that you can find in your textbook that explains in 6 steps what happened during the formation of the enzyme substrate complex. So the first thing that you have here, you have your active site, and if you see the active site is empty, and you also have your substrates, and they are going to enter the substrate enter into the active site. So here they enter into the active sites and they are held by an active site by weak interaction, forming this enzyme substrate complex. Later on, the active site can lower the energy of activation and speed up the reaction. And as a result, in your textbook, they represent the chemical reaction as a change in color. So instead of being green and red, now you can see it that is represented by a coloration, blue and yellow. So in this way, they show that the substrate are converted into products. And later on, the products are released here, and the active site is available for two new substrate molecules to go again into the active site. So again, 6 steps. to show again that enzymes are catalytic molecules and that the enzymes can use over and over.